Andreas, can you tell us a little bit about the overall architecture of the olfactory system, especially the bulb and then going to piriform cortex, just so people can think about how does information flow after it hits their nose?
Odors, so essentially any chemical that's volatile, that's in air. So I'm talking largely about mammals. It's inhaled. It hits receptors in the nose. That's receptor molecules sit on receptor neurons. There are a few hundred different types of those receptors, approximately 300-400 in humans, maybe about 1,000 in mice, but on the order of a few hundred in most mammals. And then these receptor neurons, they send their axons, so they have processes that realize information further down the line. They send these processes into the structure we've been talking about, the olfactory bulb. And they are all those receptor neurons that have the same receptor. So you can think of that code for the same kind of chemical feature. You know, if you're into chemistry, maybe it's an aldehyde group or a particular aspect of a benzene ring or something like that. Or you can think of maybe slightly simplifying of, they code for fruitiness or sweetness or some aspect of floralness or so. All these receptors, receptor neurons, they send their axons into one or a few small distinct structures in the olfactory bulb. And there they make connections, they make synapses with a couple of different types of neurons. But interestingly enough, they also already make synapses directly with those neurons that send information to other brain regions, sort of projection neurons, mitral and tufted cells. And so they send on information to, for example, the piriform cortex, which is the structure of the mammalian brain that's thought to be involved in categorizing or memorizing or recalling odor memories. But they also send their axons into different other structures, like part of the cortical amygdala, so the region dealing with, say, emotion, anxiety, things like that. They send direct connections into part of the hippocampal formation, the entorhinal cortex, possibly to do with some other aspects of memory and categorization, but also in a few other smaller substructures. And within the olfactory bulb, so this is only sort of 2 steps down from the nose, but within the olfactory bulb, there are approximately 1 million neurons in a mouse, and I think probably roughly the same in humans. And these 1 million neurons do some kind of computation. They implement some kind of algorithms that allows animals to extract specific type of information from the surrounding. And I think it's fair to say we've spent probably 20 years or so working on a variety of those local processes in the olfactory bulb. And it's fair to say that we don't really understand what kind of information they're extracting, how they allow animals to get a better picture of what the world is. But the general architecture, you have the nose, receptors in the nose, send information to the olfactory bulb. From there, it gets processed in some way and sends information to a variety of downstream areas across the brain.
Dima, is there anything you'd want to add to that?
All information obviously is on the receptor type, and you ask what we should learn if we record from the receptors. I think it's a similar, again, the analogy, if we would be able to make a transformation from the receptors to the perception, it's the same thing like we will be able to do to predict the colors from the spectrum. If we know the spectral property of individual color receptors, and from there we can predict the color. Color is the perceptual variable, and spectrum is the physical variable. The same thing here, we have the odors, that are physical variables, different odors, concentrations. And if we know the property of all receptors, then maybe that will be enough to build the transformation to the perception. In ideal situation, we can build a big table, which is boring. But maybe we'll be able to make a theory like a color, in the color thing, that we can predict perception during having the receptor property. And in this case, recording from many odors, from many receptors, is what actually also tells us how olfaction works. And one of my excitement about the new adventure with the bioelectronic nose and is that the technologies that we're developing will allow us to record not from just small number of receptors, but a huge number of receptors. Maybe I don't want again to say the exact number, but to cover with a proper brain-machine interface to connect to much larger number of receptors, and that will give us a unique information that will help us not only to solve practical questions and identify specific odors, but to understand our function also.
And if I smell like my beer or a lemon or some normal smell, is there usually one receptor in the nose that's picking that up, or does it activate all of them, or like what's generally happening when you smell a natural odor?
I usually give my students kind of an example of, one odor that activates one specific receptor, and if that receptor in a human is changed, people perceive that odor as different. They're androstenone is one example. For some people, it smells beautiful floral. Others don't smell it. Other things, it smells like urine. Beautiful example. Really not typical. Most odors engage with even single types of molecules engage tens to hundreds of receptors to varying degrees. Only the combination of these activity patterns actually encodes for a specific odor. But even more complicated, your beer, you might have a Belgian beer, I assume, that probably has a rich number of different flavors that come in it. So hundreds
exactly,
and hundreds
whereas
of different.
as German beer would be what you call a monomolecular odor in there.
Beautiful in its purity, exactly. But even any normal natural smell will have tens, hundreds, sometimes thousands of different components, which makes actually the job of a perfumist also very tricky. You put together mixtures of odors and they start smelling completely different in a still very poorly predictable.